Hello, beautiful people. Excuse me. Hello, beautiful people. Welcome back to my channel. It's your favorite teacher, Najuma Sharuk. I am here to take you through three things. Number one, we are going to look at comparing eukaryotic and prokaryotic cells. We're going to look at the structure of a prokaryotic cell and the structure of a virus. Remember, I'm here to take you through AS biology. That is the Cambridge syllabus. If you're interested in getting an A-star, if you're interested in passing with high marks, I'm here for you. Stay tuned. So we are going to start with prokaryotes. In one of our first videos, we looked at prokaryotes and eukaryotes. So I want us to remind ourselves about prokaryotes and eukaryotes. We know that all cells are categorized into two types. So they can either be prokaryotes or eukaryotes. When it comes to prokaryotes, they are placed under that category because of the fact that they do not have a nucleus. Yes, so the distinction comes from whether the cells have a nucleus or they don't have a nucleus. So prokaryotes do not have a nucleus. And whenever someone talks about prokaryotes, what should come to your mind are the bacteria. So bacteria are prokaryotes. And prokaryotes are way much smaller than eukaryotes. So since prokaryotes do not have a nucleus, then their opposites, the eukaryotes, have a nucleus. Yes, their DNA is enclosed in a nucleus. When it comes to prokaryotes, you can see in this structure of a prokaryotic cell, DNA is just floating in the cytoplasm. It is not enclosed by a nuclear membrane. So that is the major difference between prokaryotes and eukaryotes. But there are other differences that exist, but the major one is the fact that prokaryotes do not have a nucleus. So all bacteria are prokaryotes. No nucleus, DNA lies free in the cytoplasm. And then all the other kingdoms, that is the animals, the plants, the fungi, the protists, they all belong to eukaryotes, meaning they have cells whose DNA is going to be enclosed by a nucleus. So this picture shows us a typical prokaryotic cell, and it has been divided into two sections. We have a section that shows us structures that will always be present. Excuse me. Structures that will always be present in a prokaryotic cell. And then it also shows us structures that will just be found in some, not all bacteria. So when you look at structures that are always present, bacteria always have a cell wall. Similar to plant cells, they have a cell wall, only that the components differ. We saw that plant cells have cellulose in their cell walls, but when it comes to bacteria, their cell wall is made up of a peptidoglycan called murine. So this substance that makes up their cell walls is a mixture of carbohydrates, polypeptides, and amino acids, peptidoglycan. Then they have a cell surface membrane. Remember, these structures play the same roles as found in other cells. An example, the cell wall will provide protection to the inner parts of this structure of the bacterium. At the same time, it will prevent the bacterium from bursting when water enters into it by osmosis. The cell surface membrane will play the same role as in other cells, control movement in and out of a cell. They also have cytoplasm, which is just a solution of sugars and other ions dissolved in it. Then they have DNA, but their DNA is circular, which is different from eukaryotes because eukaryotes have DNA that is linear, organized into strands, double-stranded DNA. Now these ones have DNA that is circular. They also have ribosomes, only that their ribosomes are small in size, but the ribosomes play the same roles. The ribosomes will be carrying out protein synthesis. DNA will still play the same role, provide instructions for making of proteins. As a result, it will be controlling the activities of this cell. So when you look at this circular DNA, it is not organized into chromosomes like the way it happens in eukaryotes. Then this side, we have structures that are only found in some bacteria, not all. So some bacteria have a flagellum, which is used for locomotion, swimming in environments where they are found. The flagella sometimes can be more than one, or it can just be one. Then they have a capsule. In addition to the cell wall, sometimes there is an additional protective layer known as a capsule. So the capsule simply provides additional protection. That is why sometimes someone can take antibiotics and the bacterium are not killed. The bacteria are not killed simply because they have these capsules that prevent antibiotics from working on the bacteria. Some bacteria have their cell membrane infolded. So this infolding of the membrane provides an area where certain reactions can occur. Like an example, some bacteria carry out photosynthesis, but you will not find them with chloroplasts because they lack membrane-bound organelles. No chloroplasts, no mitochondria, but those chemical reactions can happen on the infolding of the cell surface membrane. I'm sure you heard about nitrogen-fixing bacteria, so they will have such processes happening on this infolded cell surface membrane. Other photosynthetic bacteria, photosynthesis will still occur on this infolded cell surface membrane. Now, some bacteria have plasmids. Plasmids are small circles of DNA. In addition to the circular DNA, we can have the small circles of DNA. Then they also have pili. Pili are used for attachment to other cell structures. Pili are like rod-like structures. You know how some bacteria can be found attached maybe on rocks or on the surfaces of different organisms. So they use these pili for attachment on those surfaces. At the same time, the pili are going to be involved in sexual reproduction. Because during reproduction, we can have what we call conjugation. We can have one bacterial cell getting close to another bacterial cell. And then this one has a plasmid, this one also has a plasmid. So they can use pili as channels through which they can exchange genes. So this plasmid will get to this other side via the pili, and then this plasmid will also get to this other side via the pili. And that is called conjugation, which is sexual reproduction. Sexual because we have two cells involved, and they will be sharing genes through their plasmids. So those are the major features of prokaryotes or bacteria. So we should be able to tell the difference between prokaryotes and eukaryotes. I've already talked about these different features: the cell wall made up of peptidoglycan. The cell provides protection and prevents the cell from bursting. We've seen the cell surface membrane controlling exchange between the cell and its environment. We have looked at the cytoplasm, and within the cytoplasm, we have seen that bacteria do not have membrane-bound organelles, so you will not find their mitochondria and chloroplasts. We've seen that their circular DNA is found within the cytoplasm. It's not enclosed. And this region where the DNA is found can be called a nucleoid region. Yeah, this term here, nucleoid region. We've seen they have ribosomes, and ribosomes carry out protein synthesis. However, for them, their ribosomes are smaller than those of eukaryotes. So they have 70 S ribosomes, whereas eukaryotes have 80 S ribosomes. We've seen they use their flagellum for swimming. We've seen that chemical reactions can happen on the cell surface membrane. They have an extra layer of protection called a capsule that prevents them from that protects them from antibiotics. The capsule also prevents the bacteria from drying out. They have plasmids. Plasmids are small circles of DNA, and these plasmids, most of the time, are very essential in providing bacteria with resistance against antibiotics. They have pili. We've looked at the pili that are used for attachments on surfaces, and pili allow transfer of genes and through plasmids from one bacterium to another during sexual reproduction, which is also called conjugation. So you're expected to compare. Remember, whenever you're told to compare, we look at similarities and differences. You're expected to compare prokaryotes and eukaryotes. So this is a summary of prokaryotes and eukaryotes in terms of comparison. Prokaryotes are believed to have existed earlier, 3.5 billion years ago. Eukaryotes came way much later. They evolved much later. Prokaryotes are always small in size. You can see their typical size is 1 to 5 micrometers in diameter, whereas eukaryotic cells, eukaryotic cells can be up to 40 micrometers in diameter. You can see that is way much smaller. Sorry, that is way much bigger compared to the prokaryotes. We've seen prokaryotes have DNA, but the DNA is circular and lies free in the cytoplasm, not surrounded by a double membrane. Eukaryotes' DNA is non-circular, it's linear, and can be organized into chromosomes, and is surrounded by a membrane, a double membrane, which we are going to call a nucleus, a nuclear envelope. These ones have smaller ribosomes, which are 70S. That is the prokaryotes. Eukaryotes have 80S ribosomes. Prokaryotes have way much fewer organelles, and they do not have membrane-bound organelles. Yeah, then eukaryotes have very many organelles, which are membrane-bound. Some are surrounded by a single membrane, like lysosomes have a single membrane surrounding them. The Golgi body, a single membrane, the vacuole, a single membrane, the endoplasmic reticulum, smooth and rough, have a single membrane surrounding them. Whereas other organelles have two membranes. We saw those organelles earlier. The nucleus, two membranes, an envelope, mitochondria, and chloroplast. They have an envelope, which simply means two membranes surrounding them. And other organelles like the microtubules, ribosomes, and centrioles have no membranes. Prokaryotes have a cell wall made up of a peptidoglycan called murine, which is a mixture of carbohydrates, those are polysaccharides, and a combination of amino acids. Then eukaryotes, yes, they have a cell wall, but not all of them have a cell wall. Like animal cells have no cell wall. However, plant cells and fungi have a cell wall, but the components differ, whereby plant cells have a cellulose cell wall, meaning they have cellulose in their walls, and then fungi have chitin in their walls. Flagella are found in prokaryotes. Even some eukaryotes have flagella and cilia, which are made up of microtubules. When it comes to the bacteria, they go through cell division by binary fission, where the cell just splits into two. And it doesn't involve use of spindle. You'll learn about this in chapter 6. Whereas this side, yes, cell division occurs by a process called mitosis or meiosis, and it involves formation of spindle microtubules. You'll learn about the details of cell division still in chapter 6. Prokaryotes, some carry out nitrogen fixation. You never find a eukaryote carrying out nitrogen fixation. So those are the major features of prokaryotes and eukaryotes. Let's look at some sample questions that you could be asked. Yes, I have a multiple choice there. Yes, feel free to read through. Which structures will be present in a cell that causes cholera? Now, we know that cells that cause cholera are bacterial cells. That is Vibrio cholerae, which is a bacterium. They're telling us circular DNA, cytoplasmic DNA, 70S ribosomes. We've seen bacteria have circular DNA, so that means one is a structure found in bacteria. And DNA is in the cytoplasm, so that is why cytoplasmic DNA qualifies. And their ribosomes are smaller, 70S ribosomes. So in that case, we can see that our most appropriate response is going to be A. So option A is the most appropriate response. Option A. Let's look at another one. They're telling us that bacterial ribosomes consist of 2 subunits and a total of 3 ribosomal RNA molecules per ribosome. An analysis of all the 70S ribosomes from a single cell of the bacterium Escherichia coli showed that there were, so here we are going to need some mathematics. They're showing us 57,000 ribosomal RNA molecules, 3 types of RNA molecules, 19,000 copies of each type of ribosomal RNA molecule. And then they're asking us how many 70S ribosomes were there in the E. coli cell. So whenever you find a question, if the question is actually not if the question, all questions, whenever you're trying to attempt them, try to read the question like 2 times so that you're able to interpret it well. Most of the time, you don't find questions that are very direct. They will have a mixture of different concepts from different topics, which you should be able to integrate so that you're able to arrive with the best answer possible. So in this case, we are having bacterial ribosomes have 2 subunits. So this is what we are looking at. A ribosome has 2 subunits. So that is our ribosome. It has a total of 3 ribosomal RNA molecules per ribosome. So each ribosome has 3 ribosomal RNA molecules. An analysis of all the 70S ribosomes from a single cell bacterium showed that, okay, so they are telling us all the ribosomes had these ribosomal RNA molecules. All the ribosomes had 3 types of RNA molecules, and then 19,000 copies of each type of RNA molecule. So how many 70S ribosomes were there in E. coli? Okay, how are you going to be able to attempt such a question? First, pause the video and try it out before I give you what I think should be done so that we get the most appropriate answer. If you don't have a calculator, pick one. You can pause the video, get a calculator so that we apply some mathematics. Okay, those that have tried out, let's see what to do. And let's find out if we have gotten the best option. Yes, we have a ribosome. They are telling us it's made up of 3 ribosomal RNA molecules. And they have given us information here. When they observed these cells from this Escherichia coli, they found out that the cell had 5700 ribosomal RNA molecules. And they're asking us how many 70S ribosomes were there in this E. coli cell. I have been told each ribosome has 3 RNA molecules. But then I have 57,000 ribosomal RNA molecules that were found in the cell. So we also have this option, 3 types of RNA molecules were found. That means that a ribosome could have any of the 3 types, but they should be 3. Yes, they should be 3 RNA molecules in a ribosome. So they are also telling us 19,000 copies of which type of mRNA molecule were found. I might not really be so much bothered about the 19 copies, but I'm going to basically use this. So I'm going to divide this by 3 because they're asking how many ribosomes. Divide 5700, divide by 3, and the most appropriate answer is going to be B. That means there were 19,000 ribosomal RNA molecules. So the answer is B. Did you get it right? Okay, let's look at some other questions. Which cell components are present in a typical prokaryotic cell? We've seen prokaryotes have 88 ribosomes. Tick is present, cross is not present. Which cell components are present in typical prokaryotic cell? Tick present, X not present. So I see ATS will be present, but not in a prokaryotic cell. So no, this can't work. Do they have cell walls? Yes, they do. All prokaryotes have a cell wall. Typical means that every prokaryote has it. Every prokaryote has a cell wall, but no ATS ribosomes. So the most appropriate answer in that case is B. So I will not even look at the other options. Then I also have this. Yes, you can read through, you can pause, read through, choose your answer before I give you my opinion. There is a theory that mitochondria and chloroplasts were originally free-living prokaryotes. We looked at the endosymbiotic theory, so this question is about that. It is thought that millions of years ago, these free-living prokaryotes were taken into larger cells by endocytosis, where instead of being digested, they became functional organelles. Which features of mitochondria and chloroplasts support this theory? That is the endosymbiotic theory. What shows that bacteria and chloroplasts were once prokaryotes? Okay, number 1, they are telling us mitochondria and chloroplasts are surrounded by double membranes. Is it part of the endosymbiotic theory? What do you think? Mitochondria and chloroplasts have small circular DNA. Yes, prokaryotes have small circular DNA. Mitochondria and chloroplasts synthesize proteins. This simply means they have their own ribosomes. So they can synthesize their own proteins. And we saw that they have 78 ribosomes. So 1 is out. This is, yes, they are surrounded by double membranes, but it doesn't really support the endosymbiotic theory. Yeah. So the most appropriate answer should have number 2 and 3. So the answer is D. The answer is D. The answer is D. Okay. Another question. You can pause and try it out still before I give you my opinion. Bacteria are found attached to epithelial cells in the intestines of mammals. Epithelial cells in the intestine of mammals. So I know mammals are animals. Obviously, they are talking about animal cells. Describe how the organization and distribution of DNA in epithelial cells differs from the organization and distribution of DNA in bacterial cells. So this question has 2 things in one. That means that for us to get the 2 marks, I should be able to describe organization of DNA and also distribution. Organization of DNA in animal cells, which are epithelial cells. How is it different from that in bacteria? And then how is distribution of DNA in epithelial cells still different from that in bacteria? Okay, we know that as far as organization is concerned, when it comes to these epithelial cells, DNA is organized into chromosomes. Or you could say their DNA is straight or linear. And it is associated with special proteins known as histone proteins. Whereas when you get to the bacteria, DNA is not organized into chromosomes and their DNA is said to be naked. Naked means that it's not surrounded by histone proteins. Yeah, they have naked DNA. And their DNA is also circular. Then when it comes to, so that is as far as organization is concerned. So any of these, you could get your mark, 1 mark. And then the other answer had to be about distribution. So distribution is where is the DNA found? So DNA is going to be found surrounded by a nucleus that is in animal cells or these epithelial cells surrounded by a nuclear membrane. Or you could say DNA is found in the nucleus, or you could say DNA is enclosed by a nuclear membrane. Or you could even say DNA is found in the nucleolus because we saw earlier that the nucleolus has DNA. So you can try out more past paper questions so that you get familiar with the questions and how to answer them. Next, let's look at viruses. Viruses are really tiny organisms. Should we even call them organisms? Because when you look at viruses, they do not belong to any kingdom. When it comes to kingdoms, we have that of animals, we have plants, we have fungi, protists, and prokaryotes, which are the bacteria. However, viruses were not assigned any kingdom because every living organism should have cells. But when it comes to the viruses, they do not even have cells. They are acellular. They are acellular. This is the spelling of the word acellular. Acellular simply means that they do not have a cell structure. No cell structure. You will not find them with cytoplasm. You will not find them with a cell surface membrane. You will not find them with a cell wall, no ribosomes, no mitochondria, no Golgi body. They do not have cell structure or structures that are found within a cell. So that makes them to qualify to be non-living because they are believed to be the link between living organisms and non-living organisms. Because non-living organisms do not have cells and viruses don't have cells, so they can be considered non-living. However, they have certain features which make them to be considered living. An example, all living organisms have genetic material. So viruses have genetic material. And their genetic material can either be DNA or RNA. DNA and RNA are nucleic acids, and you learn their details in chapter 6. Yes, you learn their details in chapter 6. So they have DNA or RNA, but not both. So a virus can either have DNA. The one that has DNA will only have DNA. Those that have RNA will only have RNA, not both. And so them having genetic material makes them to qualify to be living organisms. So when you look at the structure of a virus, it's a very simple one. It has genetic material, and that genetic material is surrounded by a protein coat. A protein coat or a capsid. This capsid is called a protein coat. And the capsid is made up of, so the whole of this green structure here is called a capsid. A capsid is made up of these subunits here, which are called capsomeres. So many capsomeres join together to form a capsid, which provides protection to that genetic material, either DNA or RNA. Now, some viruses, not all of them, some only have a capsid and DNA or genetic material or RNA. Then other viruses can have another protective layer known as an envelope. Yeah, like you can see, this is an envelope, this outside layer. This envelope is made up of phospholipids. You learn about phospholipids under biological molecules and also cell membrane and transport. So they have this envelope, and this envelope is made up of phospholipids and some proteins. Usually they get this envelope from their hosts. As they leave the cells of the hosts, they take part of the membrane, which forms an envelope that is going to enclose their capsid and genetic material. Now, all viruses are parasitic in nature. That means they cannot survive without their hosts, and they can only reproduce once inside the host. If a virus is outside of a host, it will not be able to reproduce. So when these viruses get into the host cells, they instruct the protein synthesizing machinery. They will instruct the host cell DNA to replicate so that it makes their own genetic material. They also instruct your ribosomes so that they make their proteins. So you make all these components for it. And then once the proteins are available and DNA or RNA is available, we can easily have a virus. So those structures will simply be assembled and a new viral cell, no, not cell, sorry. A new viral structure is made. So let's look at an example of a question here. They are showing us a virus, a Zika virus. This Zika virus is a pathogen that can infect human cells, and they have labeled some parts here. Obviously, when I see labels, I'm sure they might ask me to be able to write the names. Let's see what the question asks. Structure A is made up of a protein. Name structure A. Pause and try to come up with the name of structure A before I answer it. Suggest the name of structure B. They are telling us structure B is single-stranded, is a single-stranded molecule. So we have structure A and structure B. They want the name of structure A and the name of structure B. Yes, you're right. Structure A is a capsid. Structure A is a capsid made up of these many capsomeres. Okay. Structure B. Structure B, you can either write DNA or RNA. So you can still go look for past paper questions, try out more questions about prokaryotes, eukaryotes, and viruses. Otherwise, for today, we are going to stop here. Be reminded that I provide tutoring services. Feel free to contact me. I'll leave my email in the description box so that you can send me an email and we see how to link up. I'll also leave my active WhatsApp number. You can send me a message if you need my services so that we are able to get that A star. Yes. And I have told several students who have all excelled in AS biology. Don't forget to subscribe if you love the video. Share with a friend so that they are also able to learn. Yes. See you on the next one. Bye.